Okay, so in this next video, what we're going to be doing is we're going to be looking at anaerobic respiration in quite a bit more detail. And essentially, after we look at anaerobic respiration, I'm going to slightly talk about aerobic respiration. We're going to hit it in quite a bit more detail with HL and a little bit of an introduction later on. But for this one, we're mostly focusing on anaerobic and then kind of what makes aerobic respiration, what's happening here. But the first part of anaerobic respiration is important for both aerobic and anaerobic. So let me explain. Okay. So here is the food that you eat. And we're just using glucose as our example molecule. So when you eat food, let's say it's pure glucose. We have all glucose, right? And that glucose will get broken down into something called a pyruvate. Okay, and you're going to see this is part of this first step that's going to be called glycolysis. So this glucose is going to be broken down into two pyruvates. The glucose C6H12O6, right? That is a 6-carbon sugar. The pyruvate is a 3-carbon sugar. So you're going to take one glucose and you're going to snap it in half so it becomes 2 pyruvates. And the fate of the pyruvates will depend on if there's oxygen available or not, like if that's in your body or animals or fungi or any eukaryotes. If there's oxygen available, then it's going to go on and go through what's called aerobic cellular respiration. And that aerobic will make a whole bunch of ATP, okay, bunch of ATP. If there's no oxygen available because you're either using up, that oxygen too fast, and so maybe you need some additional energy, but there's just not enough oxygen to make even additional energy, then the pyruvate won't be sent to the mitochondria. It will be used to make a little bit of ATP here in the cytoplasm. But to keep making that ATP, there is a special molecule that has to be regenerated. And so what ends up happening is that pyruvate will turn into either ethanol or lactate, depending on what species you are, to be able to keep kind of producing ATP from the next pyruvate that comes around. So anaerobic is when it's low oxygen or not enough oxygen conditions to keep pumping out the ATP from the mitochondria. The count in how much ATP gets made, if it's made here through aerobic respiration in the mitochondria, you get 36 to 38 ATP out of one glucose molecule. If the fate of that ends up not going to the mitochondria and it's processed through anaerobic respiration in the cytoplasm, we end up only getting about 2 ATP. Okay, 2 net ATP. So let's take a look. We're going to be looking at this in detail. Okay, so I'm going to move myself. There we go. All right, so when we have the glucose molecule, okay, remember that glucose is going to split into 2 ATP. In that process, if it's splitting into 2 ATP, or 2 pyruvates, a glucose will split into 2 pyruvates. Okay, one glucose splits into 2 pyruvates. In doing so, the energy that gets harnessed is 2 ATP. Okay. Now, there's some other molecules that end up being made in this process. The molecules that also get made in this process are something called electron carriers. So this NADH is an electron carrier. Now this, if it were aerobic respiration, is going to be really important in making more and more ATP. But if there's not oxygen, we're not going to do aerobic respiration. So this electron carrier is just kind of dead in the water. Okay, it's not going to go on to help make more ATP. So in the process of going from glucose to 2 pyruvates, 2 ATP get made, okay, and an NADH gets made. And the NADH gets made because what happens is there's a molecule called NAD+ that is going to be reduced, meaning it's going to gain an electron and a hydrogen ion. It's going to gain an electron and hydrogen by stealing it from the glucose during that split. So as glucose splits to 2 pyruvate, an NAD ends up picking up electron and a hydrogen to become NADH. This reduction of NAD to NADH is an important thing that needs to happen to be able to get this ATP made, okay? So for this process from getting the ATP from glucose to pyruvate, to keep this going, to keep glycolysis happening, you need an endless supply of NAD pluses. okay? But if a lot of those NAD pluses get supplied from aerobic respiration because they're constantly using some of those, these NADHs get used up. But if they're not getting used up and turned back into NAD plus, well, the cell needs to make more NAD pluses. So the way it does that is it usually will transform this pyruvate, okay? So it's actually going to reduce the pyruvate, and the NADH gets what we call oxidized. So it's got to lose that electron again so it can become NAD plus. So NAD will gain an electron and a hydrogen from the glucose when it's splitting into the pyruvate and become NADH. Now that NADH has to become NAD again, and the way that it will do that is by interacting. It'll react with the pyruvate, and when it does that, it will give that hydrogen and the electrons to the pyruvate so the NAD reforms again, okay? So essentially what's going to happen here is we need the NAD plus to keep being made, and that can only happen is if we can have that NADH get rid of that electron and the hydrogen. And if there's no oxygen available, that's going to have to happen here in the cytoplasm, so it's going to react with the pyruvate. So this pyruvate now is going to be interacting with this NADH, and it essentially ends up becoming ethanol and carbon dioxide. That pyruvate gets transformed. So notice, we go from glucose to pyruvate, and then that pyruvate is going to interact with this NADH and become ethanol and carbon dioxide. This type of fermentation, so aerobic, anaerobic, this is anaerobic, no oxygen, anaerobic respiration. This, there's two types of it. This type that's carried out in bacteria and plants and many yeast will have that pyruvate get transformed into ethanol and CO2. This is why we use yeast when we're baking bread and making beer, okay? Because we essentially put yeast and sugar. So this is the organism, the yeast cells is doing the glycolysis. We put the sugar in there and it's going to break down the sugar, right? And the byproduct of breaking down the sugar is it's going to end up making ethanol and CO2. The reason why it's making the ethanol and CO2 is so it can have more NAD plus to keep that glycolysis going, okay? So that's why we get CO2, the bubbles in the beer, or those bubble pockets in the bread, okay? This is called alcohol fermentation or ethanol fermentation. We don't do ethanol fermentation in our own bodies. Animals, many bacteria and some fungi, we actually do lactic acid fermentation. So the glucose gets turned into the pyruvate, but the pyruvate, when it interacts with the NADH, instead of turning into CO2 and ethanol, that transforms into lactate, okay? So it's kind of lactic acid that you hear about. And so that continues to be made just to restore the NAD plus so glycolysis can keep happening. In us, we do that because we often need extra energy. So if you're working out really hard and you get to the point where you're needing to make more ATP than your mitochondria and all the oxygen can handle, you can make extra ATP by doing fermentation in the cell, in the cytoplasm of the cell. So we start doing this lactic acid fermentation to make extra ATP to help with power boosts, to be able to get us going past that point so we can have more than just what our mitochondria can handle. And like if we're using bacteria to say make yogurt, for example, yogurt gets kind of tart, right? It lowers the pH. Well, the pH is lowering because you're making lactic acid. Okay, and so that's why the yogurt is tart. It's because all this buildup of the lactate from doing all of this lactic acid fermentation. So this anaerobic fermentation. Okay, and as I said, our muscles do this when O2 gets low, and that extra energy and the lactate enhance muscle performance. Okay, so here's our glucose, and that is going to be turned into pyruvate through the process of glycolysis. Okay. Now, if there's no oxygen, then that pyruvate will be transformed into either ethanol and CO2 or lactate, okay? But if there's oxygen available, then that pyruvate can be transferred into the mitochondria to continue to be broken down. So this is a 6-carbon sugar. This is a 3-carbon sugar. And then it will be transported here, and all of the carbons actually end up being released as carbon dioxide, and all the energy gets harvested. Now, do you remember this molecule here, NADH, that electron carrier that we had to keep transforming back to the NAD+ in anaerobic cell respiration? Well, those NADHs that get made here and in a couple other processes in the citric acid cycle, or the Krebs cycle, so known as, all of those electron carriers actually come down and donate their electrons to this last stage. And that's where most of the ATP gets made. There's like 32 to 34 ATP made here. So you get 2 ATP here, 2 ATP in this other cycle, and then 32 to 34 here. And what's driving all this ATP production is all of these electrons that the NADH and one other electron carrier are carrying over to this last part. So you get a whole bunch more. So the big difference between anaerobic and aerobic is that in anaerobic, there's no oxygen, so that pyruvate keeps getting transformed into something else, so the NADH can keep being made into NAD+, so you can keep making the pyruvate, or keep doing glycolysis. But if there's oxygen, all that NADH, instead of being transformed to ethanol or lactate, gets sent over to the mitochondria, and that is where you end up making a whole bunch of ATP. All right, and the last little thing on this unit is just measuring the production, measuring cell respiration using a respirometer. And there's a couple of different ways. We usually would just use a CO2 probe to measure how much carbon dioxide is coming off of an organism, and we'll do that in the fall. But you can also put an organism in a chamber like this, right? And it's going to breathe in O2 and release CO2. And this chamber, if you put it under water, the water will start to go in as the organism is using oxygen. But you'd think, oh, well, why CO2 would come back out? But you use this little KOH to be able to absorb the CO2, so it turns into a solid. So as the organism keeps breathing in, the water kind of keeps coming this way, this way, this way, and you can measure how much cell respiration's been happening. There's kind of weird ethical implications to it too. But we'll do a cool one online to kind of show you what they mean by that. And then you can determine if you plot the data with X on the with time on the X-axis and volume on the Y, you can actually get a rate. Okay, I'm going to stop here because we are going to be making bread next class, so we can actually see the fermentation, the alcohol fermentation happen, because we're going to get all that CO2 to make our bread rise and get all puffy with yeast. So we can kind of see this whole process in action and talk about what's going on. All right.